I was here to talk about the poly-DP-ribose polymerase inhibitors, PARP inhibitors, and to explain how they work and how they could be combined with other chemotherapeutic agents. I think they could be all the PARP inhibitors inhibit the enzymatic activity of PARP, which is to generate polymers of poly-DP-ribose. But where they differ is some of the PARP inhibitor can poison the enzyme, and they need the cells to have the enzyme to kill the cancer cell. And those inhibitors can be used as single agents because they damage DNA. Pretty much all cancers have a lot of PARP enzyme. If you take away PARP in a cancer cell, it becomes resistant to the PARP inhibitors. But most cancers have PARP. It's an abundant enzyme. PARP trapping is the idea like you trap something when it's being active. So PARP is going through the chromatin all the time, scanning the chromatin and putting polymers. And normally it comes on and off. It's very dynamic, extremely dynamic. So trapping refers to the fact that the drugs block the enzyme when it's actually bound to chromatin. And that's what's toxic for the cancer cell. The two classes come of importance because if you just do catalytic inhibition, not having polymer being formed, then you could get very nice synergism with the certain class of drugs like the topoisomerase inhibitors, irinotecan, topotecan. You could use a PARP inhibitor that doesn't trap. It's still very synergistic, extremely synergistic. But for DNA damaging agents that produce DNA damage, like temozolomide, you do even better if you use a PARP inhibitor that induces trapping in addition to catalytic inhibition. So that's the idea with the olaparib, for instance, that associated with temozolomide. It's not better, it's just different indications. So where the alkylating agent is indicated for the tumor, like if you say glioblastoma, then certain PARP inhibitors would be optimum. If it's a tumor which is treated usually with topoisomerase 1 inhibitor, like colon cancer or ovarian cancer, then you could use veliparib and a catalytic inhibitor, and you get very nice synergy. I can report from what I hear from Ruth and in the meeting. Actually, the activity is very impressive as single agents because olaparib has just been approved. In combination therapy, the clinical trials are still ongoing, and the issue is how to manage the combinations because they tend to increase the side effects of the anti-cancer drug. So I think we still have to learn and do proper scheduling and find the right tumors to do this. I think the main point is that clearly all PARP inhibitors are alike, and there are advantages to the very active PARP trappers, which are very cytotoxic on their own, could be as single agents, but there are also great benefit to the more gentle catalytic inhibitors that could be used in combination, maybe with less side effect and toxicity. So we know more about the drugs, we could rationalize the combinations. I think on the PARP inhibitor, what we'd like to know is to have actually crystal structures of the whole enzyme, and we don't have that. So this will be basic research. In terms of the combination therapy, we have to wait for a little bit for more clinical trial to optimize. And single agents, what we can see is the BRCA tumors are very sensitive to single agent. And what we're learning is there are more and more BRCA-like, including Fanconi defects that will expand the spectrum of use of the PARP inhibitors.